Hi, my name is Will Crawsick, Scientific Services Manager at Chemical Computing Group. Today I'd like to show you how to analyze a protein-protein complex. We'll do that by looking at protein contacts, properties, and patches, and then I'll show you how we can use that information to perform virtual mutagenesis and improve the overall properties of our protein. So let's get started. So I'm just going to go ahead and use the Open button to retrieve the file that we'll be working on today from the sample mol directory. I'll select the file, hit OK, and then I'm prompted with this Load PDB file panel. This allows me to select what data we're going to load into Mo, and here I'll just accept the defaults, and now we see the structure on our screen. So this is the structure of interleukin 13. It's bound to an antibody. Antibody is shown on the left, predominantly with the yellow beta sheets, antigen on the right, predominantly with the red helices. Now, antibody candidates from affinity screening often have poor biophysical properties and/or chemical liabilities that can cause problems with respect to developability. So in the next steps, what we're going to be doing is analysing this structure that has poor solution behaviour and make an attempt to reduce the hydrophobic patches whilst maintaining affinity. But before we can get started on that, given that this is a PDB file, we first need to prepare that PDB file ready for modelling. So we'll do that using QuickPrep, the panel comprising three main sections. The first handling preparation and cleanup of the structure, second sets tethers and restraints, the third handles refinement. I'm just going to make one change to this panel. I'll explain why in a moment, and then I'll hit OK to start the calculation. I get a progress update at the top left of the main Mo window, and I just want to go back to QuickPrep so that I can explain what's going on in the background and why I toggled off the fix option. So the first stage of preparation ensures that we've got a complete model. Here we're adding in missing hydrogens, charges and side chains. If there are any missing loops in our model, we'll be modelling those in automatically for anything up to 10 residues in length. Protonate 3D optimizes the ionization states of bound molecules and their side chains. It also optimizes the placement of polar hydrogens so that we can generate a hydrogen bonding network. By default, we tether our atoms, and that's applied to the receptor atoms. You can think of a tether like an elastic band. It allows the atoms to move, but if they move too far from the original geometries, the elastic band is going to pull them back to their starting position. And this way we won't deviate too far from the original crystal structure geometries. The fix option is the one that I toggled off before running the calculation. Normally it would allow me to fix any atoms 8 angstroms or further from a ligand. This is a great idea when I'm preparing a model. It's got a ligand bound to a receptor and I want to focus in on the binding pocket. But given that we don't have a ligand, given that we don't want to fix the entire structure, that needs to be toggled off. Note that if I'm working mostly on biologics, very rarely with small molecules, I can change the default settings in QuickPrep. The final refinement step performs an energy minimization, and that ensures that we don't have any clashes in our structure. So I see the calculations complete. There's no message at the top left of the main Mo window. It tells me that our model is prepared. So next, what I'd like to do is introduce you to the sequence editor. I can launch that from the main Mo window here, and the sequence editor would be the second major window type in Mo. After the main Mo window, which we use to display atoms, we use the sequence editor to display sequences. This would also be our launching point to the various protein modelling tools that we have in Mo. Here we find that within the system, the sequence editor is arranged by 4 chains. We've got light chain, heavy chain, the antigen, and solvent. Currently, to view the sequence, I have to scroll along, so I'm just going to make a few changes to the visualization of the sequence editor, starting by wrapping our sequence. I'm also going to switch from the 3-letter amino acid code to the single letter, and I can adjust the scale if I wish here as well. Note that the sequence editor is synced to the main Mo window, so if I make a selection within the sequence editor, they become selected in the main Mo window, and vice versa. Next, I'd like to annotate our antibody, both in sequence and with the atoms simultaneously. So if I go to the sequence editor and hit annotate, you'll see some of the default themes that we have available to us, such as IMGT and Kabat. Today though, I'm going to be using CCG, and when I click that, you'll notice a few changes. Firstly, it splits our sequence into these different panels. By default, the light chain is always displayed in the leftmost panel, subunit 1. Then we see heavy chain in subunit 2, and everything else falls into the third subunit. Also note that the CDRs have been coloured so that we can see where these sit. So for the light chain, we can identify L1, L2, and L3. For the heavy chain, H1, H2, and H3 has been coloured red. That shows us that this is more variable than any of the other CDRs. Note that that colouring scheme has been extended to the mainmo window. So here we can see that we've got our light chain with the purple CDRs, L1, L2 and L3. Then we've got our heavy chain with H1, H2 and the red H3. Now you'll notice that that annotation theme has only been applied to the antibody, not the antigen. So I'm just going to go ahead, open up my system manager, and we can use this to control the rendering of our antigen. The first thing I notice is that we've got a single line for our receptor. So I'm just going to go down to the gear icon and I'm going to switch from basic view to extended, and that then provides me 3 separate chains for the receptor entry in the system manager. So the system manager provides a convenient way to be able to control the visualization of the different proteins in our system. It allows us to hide and show different atom sets. It allows us to make selections. We can also render and generate surfaces. This is quite a handy tool when I want to toggle between multiple overlaid structures. Right now though, I'm going to use the system manager to render our antigen in gold. And we also notice here that we've got a collection of sets. So if I just expand that, these collections of atoms have been created during the annotation of the antibody. So it's split the antibody into discrete sets, and that allows easy selection of these sets so that we can use these when we're running various applications across Mo. I notice in the main Mo window that we're displaying some atoms, so I'm just going to hide all atoms so that we're just displaying the ribbon graphics. And now that we've got our visualization set as we'd like, we've prepared our model, we can start to analyze these interactions. So for that, I'm going to use the protein contacts panel. That opens up, which has 3 main sections. At the top, it allows us to define the sets of atoms that we'd like to analyze. In the middle, we will soon see the detail of these contacts. Currently it's empty, we've not searched anything yet, according to the type of interaction as well. At the bottom, we can set filters. So I'm going to be using the protein contacts tool to help understand the key interactions that are driving the binding of the antigen. So for set A, I'll set that to be the antibody, and for set B, we'll use the antigen, and then I'll go ahead and click search so that it returns a list of the interacting pairs. So as an example, if we look at the first line here, we see that glutamine 1 from the heavy chain is interacting with asparagine in position 113 of the antigen. We get a readout for the energy, also the distance and where that interaction occurs. So here it's BB, whereby B represents backbone. So we've got a backbone-backbone interaction, and we've got 4 contacts. I can also see the type of interaction, and there's a key here that helps me understand that IH is ionic and H bond. Now, I appreciate I'm giving quite a lot of information here. You don't need to memorize it all. If you can't remember what BB means, if you just hover your mouse, it'll raise bubble help and that'll give more information. Or alternatively, hit the green question mark to open up the Mo manual. So the protein contacts application can be used to identify some important residue contacts. So let's have a look at some of these, and to help us analyze this interaction, it's often useful to be able to sort our data. Now we can sort by any of these column headers, and perhaps a good place to start is to sort by energy. So if I click on energy, you'll see that now our strongest interactions are at the top. So within this list, we can see that we've got some important salt bridges between negatively charged residues on the antibody and arginine and lysine residues on the antigen. If I like, I can select a couple of these and view these in the main Mo window. So I'll just isolate and center. Let me just move this panel to one side, and there we can see those interacting residues. Perhaps what we'd like to do here is just change the bond rendering so that we can understand and get an idea about the strength of the interaction. So here the cylinder view gives us an idea, because the longer the cylinder, the stronger the bond. So this provides us another way to look at these interactions, just by looking at these cylinders. But beyond the strong salt bridges, what else do we have in our protein contacts? So here we see that we've got some pretty good H bonds between residues, but by default the distance interactions have been toggled off, and that's done because often there's a lot of them. So if I just toggle those on, we see then towards the bottom of this panel quite a few of these distance type interactions. They're generally weaker, but because there's lots of them, together they become important. And perhaps within this list of weaker interactions, there could be some residues that could be optimized. However, we would need to inspect these in 3D to assess if a mutation would actually be sensible. So just a quick recap that we use the protein contacts tool to help understand the key interactions driving the binding of the antigen. But beyond this quantitative analysis, we can further analyze the interactions by generating some surfaces. So we can start by analyzing the interaction surface of our antibody. So I'll just close the protein contacts and I'll hide our antigen, and I'll just center the view on our antibody, and then what we can do is draw a surface around this protein. Now this surface is colored white, but we can map different properties onto this surface, and we can do that using the system manager. If I just click on the pull down for constant, you'll see some of these different properties that can be mapped onto that surface. So as an example, let's have a look at electrostatics. So the color coding that we see here is that red represents negatively charged patches, blue represents positive, and white is neutral. So here we see that we've got many small charged regions, but if I just rotate our antibody around slightly to where the CDRs lie, we find quite a large negatively charged region. And this is interesting because a single negative residue by itself isn't going to greatly influence the surface properties of the protein. But where we find a collection of many negatively charged residues, that can contribute to quite a large negatively charged region, which overall can influence the properties of the antibody. So it's important to take note of these. As I mentioned, we can map different properties onto this surface. So let's go and have a look at these. Perhaps what we can do is map lipophilicity onto this surface. So this will allow us to identify the greasy regions here shown in green and hydrophobic versus the purple regions which are polar. So we find that we've got a large polar patch, that corresponds to where we saw the large negatively charged patch earlier. So one would expect to see that. But interestingly, we've got quite a large hydrophobic region. And that might make us wonder, is this hydrophobic region significant? Now, to answer that question, we can use protein patches as a means to visualize the regions of interest, or regions which display an excess charge or hydrophobicity, those that are significant. So let me just go ahead and add in our protein patches. Again, the color coding is going to be the same, that red represents negative, blue for positive, green for hydrophobic. I'll just remove our protein surface just to make it easier to see the protein patches, and I'll just rotate this around slightly as well. So hydrophobic patches, such as this large one here, can be useful for protein binding. But when we get an excessively large hydrophobic patch, such as the one that we see here, that can be responsible for binding other proteins and can be a cause of self-association or aggregation. Likewise, excessively large charge patches can help binding via salt bridges, but they can also cause us issues such as viscosity. In our case, I can see that we've got both a large hydrophobic patch and a large negatively charged patch. And if I just rotate around, you see that they actually coincide with the CDRs. So they therefore sit at the antibody-antigen interface, and perhaps if I just toggle on our antigen, you can see that interaction is right over that hydrophobic patch. So therefore, it's likely to be important for binding. If I want to get further information, I can use the protein patch analyzer. So I'll just go ahead and do that, opening the patch analyzer, which gathers all of the protein patch surface information from the system. So here we've got a patch list. We've got various filtering and rendering controls. We can control the visibility of our patches, and we can identify specific residues associated with a given patch. Here we find that the patches are arranged by size, so I can quickly identify that the largest patch with excess properties is our hydrophobic patch. And this may be responsible for solubility issues. If I just move this to one side, we can have a closer inspection by firstly selecting that patch, and then what I'd like to do is have a look to see where these residues are in our sequence editor. So what I'll do is I'll just isolate that one patch of interest, and then I'll color this in the sequence editor. If I click on the sequence editor, I can see that green residues here now appear in the sequence editor corresponding to that hydrophobic patch. And I can see that there's generally quite a big overlap with the CDR regions. So I know from this that the hydrophobic patch sits in the CDR region, and that becomes especially important in the H3 loop, which is more variable. Beyond a sequence analysis, I can also analyze this in 3D. So if I just go back to the patch analyzer, and what we can do is if I isolate the residues and center that, we'll be able to see this in 3D. Perhaps what I'd like to do is just change the transparency slightly of that patch so that we can see the residues underneath. And remembering the goal of improving the solubility issues, we need to consider a means of breaking up this large hydrophobic patch to prevent aggregation. So these residues are potential candidates for mutagenesis. I'll just close that panel. But beyond the visual inspection, we can also use protein properties to calculate a wide variety of biophysical properties, which will allow us to further assess developability and identify potential liabilities. So let's have a look at our protein properties. The panel comprising three main sections. At the top, we can specify a means to view and calculate these different properties. We can also specify the environment in terms of pH, temperature, salt concentration. We also have this sample option that allows us to explore different conformations and protonation states of the system. In the middle, we list the properties, and the very last section allows us to filter and visualize. So you'll notice that we've got two main modes to this panel. We've got calculation mode and viewing mode. The panel will switch to viewing mode once we have some results to view. So I'm going to leave the pH, the temperature, and the salt concentration at the defaults, but I'm going to be calculating the properties of the antibody. Sample would allow me to consider different conformation and protonation states. I won't be doing that today though. And in the middle here we see a list of just over 100 descriptors. The 28 most commonly used descriptors have been selected by default, and these are all populated based on their relevance to protein developability. So in this list, you'll find things like charge regions, various molecular and residue properties. So I'm just going to go ahead and run that calculation, I get a message at the top left of the MAMO window that tells me that that calculation has been initiated. As soon as the calculation is complete, it automatically switches across to the property viewer where I can have a look at the results. So here I can find that we've got the various areas of charge patches, we've got various mass, surface and shape descriptors. I notice that we've got one interesting descriptor here in light of the analysis we've just been doing. That relates to the area of hydrophobic patches near the CDRs. So again, more useful on that more useful information on that hydrophobic region. Where I see a plus here, that represents that we've got residue descriptors, and I can expand on this. So if I just click the plus, this then displays a list of all of the residues and their associated. In this case, I clicked on ASA, which is accessible surface area. So I get a readout for each residue and their associated accessible surface area in Armstrong squared. However, I often find that to get a good view of all of the residue information, I can toggle on Residue Table, and that gives me a complete list of all of the residues and their associated descriptors. So next, what I'd like to do is consider looking for other potential liability issues with our antibody. We know that certain amino acids can become unstable with time. Methionine, for example, can oxidize over time if exposed to solvent. So perhaps one descriptor of interest would be to assess if methionines are exposed. So again, using the residue table and sorting by the headers, I can sort by the residue exposure, and then I can sort by the residue name itself, and that will allow me then to scroll down until I find my methionines. And here I see that we've got 3 methionines. So the first 2 are not exposed at all, but this third one is actually 66% exposed. So that could be a potential liability. So I'll just select in Mo that residue, and I see that it's become selected just here, and I'll close the panel. So there's our selected methionine in position 92, and I can see that it sits in a CDR light chain, which tells me that if this did become oxidized, it could impact the affinity with the antigen. It also sits in the hydrophobic patch, which is interesting. So just a quick recap, that we found a residue that could be linked to liability. It also happens to be hydrophobic and therefore could affect solubility. So for these reasons, we might want to mutate this methionine to another residue. The question then becomes, to which residue do we want to mutate? And to answer that question, perhaps the way to do this best is to bring back the antigen so that we can see what our methionine is interacting with. So I'll just bring back our antigen, and then what I'm going to do with the methionine selected is hit Site View to isolate that. And I'm also going to hide the patch now that we know that it's in the hydrophobic patch. And we can view that. So I'm going to want to mutate this residue to something that's not hydrophobic in order to help break up that hydrophobic region. Of interest, I can see 2 bonds here. So this is important. I'm going to want to keep or even improve upon these interactions. I certainly, at this point, don't want to negatively impact the affinity or any other property. So I'm looking for something of a similar shape and size. And for that reason, glutamine comes to mind as a suitable candidate for mutagenesis. At this point, I think we can say that we've finished analysing the complex and we're now ready to perform our virtual mutagenesis. To perform the virtual mutagenesis on a single amino acid, I can use the protein builder. We see that pinned to the MAMO window, and we can use this to make interactive mutations or to build novel chains. The panel itself comprises 3 main sections. At the top, a list of the standard amino acids. We also have a list of the non-natural, plus options to be able to customize our own. In the middle, we're able to explore conformations and perform minimizations. At the bottom, we can explore rotamers. Now here I already have the methionine selected. As a result, it's highlighted in blue within the builder. The builder itself is set to mutate mode. It's not prepend, it's not append. So to make the mutation, I simply click the desired residue and we see that mutation happen in real time. Next though, our mutated residue needs to be remodeled within its local environment. So a standard workflow for this would be to repack the sidechain with the environment fixed. That allows us to find the lowest rotamer energy, and then I can minimize. There are a few options. I'm going to minimize with the selected sidechains only, to minimize the mutation site with the sidechain free to move and the environment restrained. Another option here would be to minimize the selected residues with the entire residue free to move, which allows assessing backbone movement of a loop. So here I see that we've got a H bond, which is encouraging. It suggests that we're maintaining some sort of interaction with the antigen. But let's have a look at the impact of this mutation upon the surfaces that we generated earlier. So I'm just going to bring back the ribbon for the entire structure. I'll hide the antigen itself, and then we can bring back the surface. So this is the protein patch as calculated before we made the mutation. So next what I'm going to do is regenerate the protein patch, this time considering the mutated residue. And we see reassuringly that that hydrophobic patch is now greatly reduced in size, which is exactly what we were hoping to see. If I just select that residue, we can bring back the antigen, and I'll just click Site View to isolate that specific interaction. So by making this mutation from methionine to glutamine, we see that we reduce the size of the hydrophobic patch area. We're also picking up a hydrogen bond with the antigen to maintain an interaction. And we see that the protein builder tool is a good approach when we want to mutate a single amino acid and we know what it is that we want to mutate to. But suppose that I want to mutate multiple positions, or perhaps I don't know what it is that I want to mutate to. For that, I can use the protein design application. Another benefit of using protein design is that we can also calculate affinity and stability. And that's quite important. In this case, we see that we've got a H bond, but we don't actually know whether the overall affinity has been improved or whether we expect to see a decrease in affinity. So what I'm going to do next is show you how to make this same mutation again, but this time using protein design. So I'm just going to use the undo function in mode to go back in time before we made the mutation. I can use the edit undo or Control Z on the keyboard. So I'm just going to go back, Control Z until I get back to the point just before the mutation. And what we'll do then is close the builder and repeat this same mutation, this time using the protein design application. So within the umbrella of protein design, there are different operations that can be performed. I'm going to be using residue scan, which opens our panel, and you'll notice that those same operations appear here on the left of the panel. At the top, I can specify my structure or a database of structures. In the middle, I can select the residues for mutation, together with options to customize which mutations I'd like to perform. And at the bottom of the panel, that allows for calculating the impact of my virtual mutants upon affinity and protein properties. Here I can also sample different conformations and protonation states using the ensemble option. So here I can manually just type and change the amino acids that we want to mutate to. However, I find it quite useful to use this notepad icon. So here I can select the standard amino acids from a list. Below that sits the non-natural amino acids. But if you recall, one of the benefits of using the protein design application is that we can make multiple mutations. In this case, what I'll do is use the bottom part of this panel, which allows me to select the mutations based on their properties. Now, in this case, we want to break up the hydrophobic patch. So for that reason, I'm going to click on the polar residues. So we see those polar residues appear here in the panel. And next, remembering that another benefit of using protein design is that we can measure affinity. So I'll select affinity relative to the antigen so that that's written to the output database. Notice that the protein properties is toggled on, so we're going to be calculating our protein properties and our patch properties. So I'll just go ahead and click Build to initiate that calculation. And I'd just like to go back to Residue Scan and just explain a little more specifically about this site limit. So site limit allows us to control how many sites are mutated simultaneously. Remembering that we can mutate multiple sites and we can make multiple mutations per site, we've got potential to generate thousands of mutations using this residue scan application. This can become computationally intensive, so for that reason I might want to consider making use of HPC or cloud resources. If I wanted to do that, I'd set up the panel exactly as I've just done, but I'd be using the batch button. So what that would do is take the run file as I've specified here and allow us to send that run file directly to the cloud or a queuing system. I'd also like to take a moment just to run through the different operations that we can perform under the umbrella of protein design. So starting with alanine scanning. We can use this to systematically explore affinity. In this mode, the selected residues will be mutated to alanine, the smallest residue that has a side chain, and then we can see what effect this has upon affinity and if the site could be a viable target for mutations. The mutations are done sequentially in a single point mutation fashion. Disulfide scan is used to identify pairs of residues that could be mutated to cysteine so that we can create new disulfide bonds. So this is a useful application when we want to increase the stability of our protein. Residue scan produces single point mutations of the selected residues that are prone to natural mutation. So this is leveraging our knowledge on SNPs. Residue scan is what we're currently working on at the moment, so I'll skip straight past that to sample sequence, which randomly searches the sequence space as opposed to enumerating all of the possibilities as we do in residue scanning. So this provides us the ability to search and optimize a larger sequence space in conjunction with mutation analysis by performing statistics on the search space. And then finally, we've got sequence design, which performs an optimized optimization of the selected sequence by systematically mutating each residue, and it allows mutations to compete with each other for a specific site instead of producing all possible mutations. So then the best scoring mutations per mutation site are displayed in the output. Speaking of output, we'll have a look at the database viewer where our results are being written to. The database viewer would be the third major window type in Mo, after the main Mo window and after the sequence editor. It looks and feels much like an Excel spreadsheet, but if I drag this field open, you can see that it actually contains the 3D structure of our molecules. From here, we can perform different types of calculations. So for example, I could jump into protein properties from here if I wanted. So the residue scan application is repacking the side chains of the mutated residue and the environment. It minimizes the mutation site, it adds the model to this database and calculates the scores. You'll notice that the first line here in terms of mutation is effectively self to self, and the reason that we've done that is so that we can establish a baseline to which all of the other mutations can be compared. So this is particularly powerful when we start thinking about affinity and stability, because I jump straight to delta affinity and delta stability, which are measured relative to that self-to-self wild type. If I just scroll along, you'll see that we're also calculating the protein properties as we saw earlier in the panel as we went through that. And along with this, we see some interesting results. So here we've got dpro patch hide. So this is the size of the hydrophobic patches near the CDRs, and it's reassuring to see that these are negative values, suggesting that the mutations that we're making are actually reducing the size of the hydrophobic patch near the CDRs. Now, I could use our database viewer to sort according to this column, but in this case I've only got a handful of results. So for that reason I'm going to go ahead and browse so that I can have a look at these mutants within the 3D environment while also examining the quantitative data. If I just move this to one side. So we see here that we've got our mutated residue shown in green. It's relative to the co-crystal structure, but I do have an option to toggle on the wild type. Within the browse panel, I get my delta readouts, which I find particularly useful, one for stability, one for affinity, and I can add other fields from the database viewer to this browse panel. So for example, I can keep an eye on the area of the hydrophobic patches next to the CDRs. So from here, this is our self-to-self mutation, and then I can click through and have a look at the impact of some of these other mutations that we performed whilst monitoring affinity and the size of the hydrophobic patch. So that brings us to the end of today's session, where we explored the protein engineering tools in Mo. This workflow allows us to prioritize experiments based on the computational findings. Today, we analysed an antibody-antigen complex by calculating surfaces, patches, and finally properties. And then we performed a virtual mutagenesis. First using the protein builder and then using protein design, the latter allowing us to keep track of changes in affinity and stability while improving solubility and removing the potential liability. I hope you've learned a lot today. If you have any questions, please do reach out to me at support@chemcomp.com.